Hello, everyone. My name is Ziv Bar-Joseph, and I'm a professor in the Computational Biology and Machine Learning department at Carnegie Mellon University. First of all, I wanted to welcome everybody to this research talk. Unfortunately, you will not be able to ask questions during the talk. However, feel free to email me if you have any questions or comments after you finished watching. What I would like to talk about today is some of the work that we have done related to modeling viral infection and response networks that may be relevant to some research focused on the impacts and what the novel coronavirus is doing, which unfortunately is something that the entire world has to deal with nowadays. Our interest and our focus on this area is not on the sequence information that the virus encodes, but rather on the functional genomics, or the way the virus functions once it enters the host. Several years ago, we developed computational methods for integrating gene expression data or other type of time series data with static data. Static data is data that doesn't change over time. For example, the sequence of an organism, our sequence, motif information, whereas time series data is data that can change over time. For example, the levels of genes in cells, the level of the viral proteins inside cells, epigenetics, and so forth. Our method basically integrates the time series data and the static interaction data in order to reconstruct what we call a dynamic regulatory map. This map provides information on what are the major profiles or the major groups of genes that respond to some event, and how these are being controlled or regulated primarily by transcription factors, but also by other proteins. The method is based on a computational model or a computational technique, which is known as input-output hidden Markov models. Many of you have heard about hidden Markov models. These are very famous subtype of a general family of methods known as graphical models. And in hidden Markov models, we have something called the hidden layer, which is a layer that encodes information that we don't observe. And then we have the observed layer, which is the things that we observe. And our goal is to infer what is the things that are hidden. In our case, we represent the gene expression levels, the information that we observe about genes, as the observed level, and we are trying to infer the regulation, why some gene is expressed at a certain time point. And the explanation that the model provides is because some transcription factor can regulate it. The input layer is static information, or basically it encodes a static information about which transcription factor can regulate which genes. Now, not all transcription factors are active in all states, and they don't always regulate all the genes. So this is information that is independent of the context of the case where the study was taking place, but can provide background or baseline for us to know which transcription factors are active. Input-output hidden Markov model basically extends regular hidden Markov model by changing the transition probability, so the emission probability stays the same, but the transition probability now depends not just on the hidden states, but also on the input or static state. This method we call DRAM, and we applied it very broadly to model a lot of different types of data, including several responses, several differentiation network. More recently, this is not shown on this slide, but a lot of the work we have been doing with this method is focused on something called single cell analysis, so data within single cells, and the method can be extended to model that as well. But for our main focus here on viral infection, the method that combines gene expression and transcription factor misses a key component. The current models that we develop represent the way genes are activated or repressed following some event, and also the transcription factor that regulates them. But it doesn't tell you what actually drove this transcription factor to regulate them. Some transcription factor react directly to the response, to the stress, for example, to the virus, but a lot of transcription factors are only regulated by signaling networks. Signaling networks are networks that contain a group of genes that one controls the other until they activate the transcription factors that then activate the gene expression. So a key question for us is, who is controlling these regulators, or the master regulator, the first responders, in order to activate gene expression? And the answer in many cases is that these are regulated by signaling networks that are activated by receptors that basically observe or are activated by the viral itself. So for example, when something binds to the cell, like the coronavirus, which enters the cells using its spike proteins, this activates basically some type of a response within the cell, because the virus then acts goes inside the cell, and then a lot of the viral genes are being first of all transcribed, then translated, and these interact with a lot of the host genes to activate the response. And the question for us is, what happens after the virus enters the cell? What are the genes that the virus interacts with within the cell, and what do these genes do to activate the response that the virus wants? If we can block those, we may be able to block not the entrance of the virus to the cell, but at least the impact it has on cells, and in this way, perhaps cure some of the impact that it basically does to the cell. Okay, so this is the question. And in order to answer that, we need to combine different types of data. One is the interactions that the virus proteins have with the host proteins, and then the interactions that host proteins have with themselves, and then interactions that host proteins have with transcription factors, which drive gene expression. And to do that, we learn two types of networks. One network is a network that goes from the viral proteins to the transcription factors in the cell, and these go through a lot of different layers. And then there is the network that we have just shown you that controls the response of the cell using gene expression to these initial signals from the virus. And to do that, we iterate on two different processes, or two different methods. One identifies the transcription factors that activate genes, as I showed before, and the other tries to link these transcription factors to the sources, to the viral proteins that are activating them. And this is the basic idea. We have a set of viral proteins, or viral proteins interacting with receptors. These go through several protein-protein interaction within the cell, finally reaching a transcription factor which activates the genes that we see. So if we have information on which protein in the virus interact with which host proteins, and then we can combine that with information of which host proteins interact with which other host proteins, and then we combine that with gene expression coming from infection studies on humans, on mice, we can somehow, or we hope to infer the pathways that the viral infection hijacks in order to basically replicate inside host cells. So we have done that for a few, or we tested that on a few basic simple, I would say, infections. The first is just the common flu, H1N1, which is basically the common flu. And this is the type of network that we get. In this network, we see genes that are expressed in human in response to the flu, and the transcription factors that are activating them. A lot of these are known immune response factors. So of course, humans activate some innate immunity, but there are also some other transcription factors that activate replication and so forth. So this is the response network, as I've shown you before, of transcription factor activating genes. But we can also get the network that the viral gene uses in order to activate these transcription factors. So what I'm showing here in red are proteins that directly interact with the flu virus proteins. Similarly, for the coronavirus, there are other host proteins that are known to interact with coronaviruses. These are overlapping but different than proteins that interact with the flu. These red proteins that interact directly activate the blue proteins, which do not directly interact, but indirectly mediate the viral impact to activate the green ones, which are transcription factors that then drive gene expression and the host basically response to the virus. Now, if we can analyze this network, we might actually find bottlenecks. These are proteins that if we remove them, the virus impact may be diminished. And we have done some of this analysis, and people have done experiments something called single knockout or double knockout. And you can show that some of these, especially the ones that are shown here in diamond, are actually able to reduce the viral load in cells if they are knocked out. So this validates that some of the key proteins that the network finds indeed are critical to the ability of the virus to replicate or to impact the host. And you can do several other analysis algorithms. This was one method that we developed that was based on graphical models. We have also extended that to integer programming method, and I'm not going to the math. We don't have a lot of time. The research talks are pretty short, but the idea is to find a set of proteins that together explain all of the impact that we see going from the viral genes to the gene expression. And this method, this IP method, we apply to HIV data, another viral infection that is known to impact host and gene expression. And in this case, we looked not just for potential targets for drugs that can treat human proteins in order to reduce the viral infection, but also to try to understand if we can determine not just which proteins to target, but also when to target them. So for example, if someone comes very early in the disease, they might need different treatment than someone who comes slightly later, or someone who comes, unfortunately, at the very tough and rough stages of the disease. Now, again, in this case, this is based on some infection data in 24 hours, but in reality, this can extend to much longer time frames, and it can also be adapted to different cases or different populations. For example, people with background conditions, older people, and so on. This is the network that we found. Very similar idea, although slightly different representation. The red proteins are the proteins that interact directly with the HIV protein. The blue proteins are these intermediate signaling pathways, and the green proteins are the transcription pathways. Based on this, and these happens in different stages. This is the first stage, the second stage, and the third stage that we predict. Based on this, we actually predicted a bunch of proteins that if we treat them with a specific drug that downregulates them or knocks them out, can reduce the viral load. And we had a ranking for the proteins, and based on this ranking, we selected a bunch for which drugs were known. And once you treated these proteins with the drugs, and this lists the drugs that were known for some of these proteins that we predicted and the predicted phase for these, once you treated them, you saw that not always, we didn't get 100% right, but for several of the proteins we predicted, if you treated the cells with this protein at the right time point we predicted. So for example, in this case, we predicted it to only act initially, but not later. And indeed, if you treated it early, it helped. If you treated it later, it helped, but not as much. Similarly here, if you treated it early, it helped, but if you treated it later, it did not help as much. So this shows that by modeling the way viruses interact with hosts through the signaling network and the transcription factor network, we can find potential targets, potential human proteins that we can target by drugs. And then using those, or by targeting those, we can reduce, maybe not eliminate, sometimes very close to eliminating, but reduce the impacts of the virus on the host. Now, taking this was applied, as I said, to flu and HIV. If we want to apply it to coronavirus, we can use a similar strategy. However, we need more data, because right now we don't have a lot of information on what proteins in the coronavirus interact with the host, although we may be able to get it from similar proteins. We don't have information on what gene expression patterns appear once the coronavirus enters the host. And again, this can be done, but right now, there's still not a lot of information on that. But once these two things are provided to us, we can use the same set of modeling techniques, maybe we will need to extend them, in order to identify potential host proteins, maybe even subdivide those into different potential groups. And that's it for this presentation. As I said, I'm more than happy to discuss if you have any questions about this, and I will see all of you again in the Q&A on Sunday. See you.